All right, this video is going to cover kidney anatomy and renal physiology, and the notes aren't incredibly long. There's not a ton of information here, but some of the concepts are actually fairly complicated and draw together concepts from cardio as well, because there's a lot going on with managing blood volume and blood pressure and things like that. So I'll try to be thorough and really emphasize the conceptual stuff that's going to be a big part of the exam. So, intro slide. These concepts of what the kidney is trying to accomplish. Fluid volume. This is primarily a concern for us because it has an effect on mean arterial pressure. So, total, fluid volume in the entire body, and especially blood volume, affects systemic pressure. And that's something that should be kept at homeostasis at all times. And that responsibility does primarily lie on the kidney, because your kidney is constantly taking fluid out of the blood, filtering it. In this case, the kidney really is a filter, unlike the liver. And then deciding, does that water go back into the blood, or does it get excreted as urine? And if you check out this number, kidneys on average for an average sized person will filter about 180 liters of fluid a day. That number we call glomerular filtration rate, right, or GFR, and you're going to see that number a lot. It's not a very good G, I suppose. So GFR, the amount of fluid that the kidneys actually take out of the blood, filter, and then you can see, just sort of by definition, if you're taking 180 liters a day out of your plasma, for an average person, maybe 177 of that is going to go right back into the blood, one way or another, and maybe about 3 of that is going to leave the kidney as urine. A normal person is not forming more than 3, 4, maybe 5 at the most if they're drinking a lot of water, liters of urine a day. And the question is, why do you have to do so much filtering? Right? If you're only forming 3 liters worth of urine, what's the point of filtering 180 liters of fluid? And primarily what it comes down to is you have to keep that GFR number high, because your kidneys are constantly sampling substances from your blood. And I want to make a list. That list we'll call, I'm going to abbreviate here, categories of molecules. We'll say categories of molecules filtered by the kidney. The first one we'll call caloric nutrients. The second one we'll say non-caloric nutrients. And then the third category we'll say toxins / waste products, like metabolic waste. And your kidney has different rules for these things. For caloric nutrients, example, glucose, amino acids would also fit in here. The rule for things like glucose is your kidney should return as close as you can possibly get to 100% of that glucose back to the blood. The reason being, if you have extra glucose, obviously you don't want to eliminate it in the urine, because that's wasting calories. You use insulin to store that glucose as glycogen or as fat or things like that. And so if it has calories, it should never wind up in the urine. That will happen in diabetics that have severe hyperglycemia, and I'll get more into that a little bit later. But in a healthy person, that should never happen. On the other extreme, toxins and waste products, there the goal is eliminate as close as you possibly can to 100% of those things in the urine. And so those two are simple, and they don't really require regulation, right? Glucose, just pump as much of it as you can back into the blood, because that glucose gets into the filtrate. For reasons that we'll get to in a little bit, you can't stop glucose from leaking out of your blood and into the filtrate. Filtrate is just the word for the fluid that the kidney has in it after water has left the blood. And so you got to pump it back in. And so active transport, much like the small intestine does to get glucose out of your food, the kidney's using a similar mechanism to pump that glucose back in your blood so it doesn't wind up in the urine. All right, this middle category, non-caloric nutrients, these would be things like minerals like sodium and potassium and, other minerals as well, chloride, magnesium, etc. Vitamins would fit that category. A really big one that maybe gets overlooked is water. Okay, these are all things that you can't really store extra of. If you drink extra water, extra sodium, extra vitamin C, whatever, there's no storage mechanism for those things. The one exception is fat-soluble vitamins you can store a little bit of. But for the most part, if you take in too much of these things, which you normally do if you have a halfway decent diet, your kidney has to try to maintain homeostasis. And so we'll say for these, the rule is retain enough for homeostasis. And then we'll say eliminate the excess. Okay. That means that this category has to have control mechanisms. And homeostasis always requires negative feedback. And so for all these things, and we're not going to go through all of them, we'll do the ones for water and for sodium and calcium and maybe a couple examples beyond that. But you have to have some sort of negative feedback mechanism to make that work. Okay? These two are simple, really important, but very simple. This is where the complicated stuff is. And that's why that number has to be so high. One of the reasons is you're constantly sampling your blood to figure out if you're maintaining homeostatic levels of all those nutrients. And if not, then obviously something has to be adjusted. Okay? pH can also get put in that category because hydrogen ions and bicarb both fit in the category of non-caloric nutrients, and those are also regulated by the kidney. That's more of a long-term thing than a short-term thing, unlike blood buffering that we just talked about. Okay, and then blood pressure is directly related to blood volume, and so that makes sense. All right, some quick anatomy. This is pretty straightforward. The kidney sits in the lower posterior thoracic cavity, so if you read the labeling, that's L2. That's about the center of the kidney on the vertical axis. And obviously blood flow coming in, renal artery in, renal vein out, standard systemic circuit circulation. And the one thing is the kidneys aren't very protected. Those lumbar vertebrae don't have ribs, and so the kidneys are exposed just below your lower ribs if there was an impact. And that's one of the reasons why you have a pretty substantial layer of fat surrounding the kidney. It's not there for calories or for any nutrient reason. It's entirely a matter of physically protecting the kidney like a cushion if there's some sort of impact. Okay, kidney and cross-section. So the terms up here you should be familiar with, just so that you can describe the movement of fluid throughout the kidney. And the gist of this is, obviously blood is coming in that renal artery, blood's coming out that renal vein, and along the way there is some filtering going on. And we can't see those filters now because they're microscopic, and we'll get there a couple slides ahead. But for right now, the gist is this area up here that you call renal cortex, that's really where the filtering is happening. And it's all happening on a scale that's microscopic and really, really small. But as an analogy, the way that I think of it anyway is these structures down here, called renal pyramids, which terminology-wise, this whole region in here is renal medulla, and then each of those structures within it is one pyramid. Think of those pyramids as being kind of like filters in a way. The filtering really mostly happens in the cortex, but it's almost like you are filtering fluid Right? These are water droplets out in the cortex, and those water droplets are all being poured into that renal pyramid. Kind of looks like the shape of a coffee filter. And if that filtrate is dripping down through little tubules in the renal pyramid, you really have two options. Option one is that water gets taken back out and sent back into the renal vein just to put it right back into circulation. The other option is some of that water leaks down through the bottom of the renal pyramid. These openings on the bottom of the pyramid, each one is called a calyx. So the direct opening off the bottom of one renal pyramid is a minor calyx, and the larger collecting area that they then pool into, you call a major calyx, and all the major calyces connect to this big open area here called the renal pelvis that empties into the ureter, and the ureter will eventually connect to the urinary bladder. Okay, but once filtrate comes out the bottom of that renal pyramid, it's gone now. It's urine. You can't get it back. And so if you want that water back, it has to be taken out in the medulla, in those renal pyramids, to get it back into the renal vein, into circulation. And again, the vast majority of fluid does end up there, because if you have 180 liters being filtered throughout both kidneys, again, 90-something% of that, 95 or more percent of that, is going to wind up back in the renal vein and being reabsorbed. And only a very small percentage will wind up coming out those calyces and into the ureter. All right. Okay, little quick histology. Urinary bladder and ureter. So this is ureter. All of this tissue is smooth muscle. And that's really the bulk of the organ. A bunch of smooth muscle. You've got a little bit of connective tissue here in lamina propria. And then this very dark purple here is a transitional epithelium. If you remember from AMP1, transitional epithelia can change their shape when the organ expands or contracts. And so the gist is the ureter has all that smooth muscle because it is actively pushing fluid, urine, from the kidney to the bladder. And the reason for that is you don't ever want backflow of urine from the bladder to the kidney. That starts putting you at risk for kidney infections. And so that muscle stays clamped down very, very tight and has to push fluid down. And the squeezing of that muscle, even when you're relatively at rest, makes sure that fluid never goes backwards and gets from the bladder back up into the kidney. Otherwise, you would just pressurize urine back up into the renal pelvis and eventually back up into the pyramids, and that would be just catastrophic for kidney function. Okay. Bladder is very similar. This is transitional epithelium. That's some connective tissue in lamina propria, and then a very, very thick layer of smooth muscle. Again, because obviously the bladder has to squeeze to pressurize urine to actually force it up the urethra when it's time to empty the bladder. Okay. And this picture just gives you a bit of a visual. This is an X-ray done with contrast, and you can see that's the bladder down there. This is the outline of the renal pelvis and the major and minor calyces. So my red line is tracing each of those would be a minor calyx, and then the entire kidney probably looks, something that was terrible, but something like that on either side. Okay, and then you can see the ureter connecting the two, which is actually a fairly long tube. For reference point, that's L5, right? 4, 3, 2, 1. Get the idea. All right, renal blood flow. I'm not going to spend a whole lot of time worrying about this, but the thing that I like about this picture is it shows you that all the capillaries, right, branching off the renal artery and eventually becoming the renal vein, the bulk of them are in the cortex. Those capillaries effectively are the filter, and you'll see the structure of it before too long. Okay, but that's where fluid exchange is actually happening, where you'll have water, some of it anyway, leaving the blood, and the tiny little holes in those capillaries are effectively what's serving as the filter, because it really is a structural filter in that way. All right, this slide is showing you illustrations of microanatomy of the kidney, and this is really, really important because this structure, this thing is technically called a renal corpuscle. Everyone just calls it a glomerulus because the ball of capillaries inside of it, right, so this arteriole coming in swirls around inside of there and eventually comes back out. That ball of capillaries is the filter of the kidney. It's called a glomerulus. If you combine that with the connective tissue capsule around it, you technically call that a renal corpuscle. No one ever uses that term. Everyone just says glomerulus. That structure plus this network of tubes that it connects to, that whole thing is a nephron. And the nephron is effectively the functional unit of the kidney, so to speak, because your kidney is really just a couple million nephrons between the two, all working together. And each nephron is a microscopic structure that can filter a tiny little bit of fluid, but when you add up, about 1 million per kidney, you can get a pretty big volume of fluid filtration going on. All right, so let's go through parts. Just as this blood vessel in red would be a branch off of the renal artery. You see here they're illustrating everything up here would be in the cortex, everything down here would be in the medulla, and so the glomerulus, and then these two sections of the tube, the proximal and distal convoluted tubules. This one's proximal because it's close to the glomerulus. This one's distal because it's further away, and convoluted just because they zigzag all over the place. Those are all cortical structures. This loop down here, you'll see it called nephron loop, renal loop, sometimes the loop of Henle after the guy that discovered it, all the same thing. That's all in the medulla. So if you're looking back on this page, cortex out here is where you would find glomeruli and PCT, DCT, and then the medulla, those renal pyramids are where you would find the loop. All right, so the goal in all of this is filter fluid out of the blood, right, that's coming in that renal artery. So inside of those capillaries, there's a little bit of water that's going to leak out. I think of that glomerulus as being very much like the head of a garden hose nozzle where water's spraying out. The catch is we put that connective tissue capsule, right, that glomerular capsule, surrounding the glomerulus so that all of the water that gets filtered out gets pushed into the renal tubule. Okay, so this entire structure you call the renal tubule, and then like I said, we break it into sections. That's PCT. The loop is split into segments. There's a descending limb, descending meaning just going down, and then you go around the loop and back up, so that's the ascending limb because you're ascending back up toward the cortex. And then that fluid will move through the DCT and eventually into a larger structure called a collecting duct. A collecting duct has openings to lots of nephrons, and so maybe 10 or 15 different nephrons connect to a single collecting duct. All right. What's happening along the way here is that renal tubule is primarily where fluid is being taken back out of the renal filtrate. Because remember, in the blood, you would call the fluid blood or plasma. As soon as fluid leaves the blood and starts moving through the renal tubule, you start calling it filtrate. Right? And the total amount of filtrate, remember, that's your GFR. Okay, so if you add up all the nephrons in both kidneys, and we're filtering, that ballpark 180 liters a day number out of here, only about 3 liters of that is winding up as urine, because if fluid leaves the collecting duct, it's coming out as urine. That collecting duct would drop out of the bottom of the renal pyramid, effectively into a minor calyx. So the other 177 are being drawn back out of the renal tubule all along the way, right, and out of the collecting duct too, in some cases. All of that reabsorption is eventually taking fluid back into the renal vein, so you can keep your blood volume up. All right, so this is a zoomed-in view of what's going on in a glomerulus. Right. So this guy, afferent arteriole, leads into the glomerulus. At that point, it's just a swirling ball of capillaries. Fluid moves around. Some of that fluid is going to leave the glomerulus and enter the glomerular capsule, and then eventually the renal tubule. Remember, the first leg is that PCT, proximal convoluted tubule. Whatever doesn't get taken out as filtrate is going to leave through the efferent arteriole and eventually make its way back to the renal vein and get back into circulation. Right, so this is your actual filter, right? The physical filter of the kidney is just tiny openings, pores, they're called fenestrations, in the walls of those capillaries, and it's just a size thing. Very, very small molecules like glucose and minerals and amino acids get through, and large molecules and whole cells, for the most part, don't. Okay? And so the cutoff line is really like individual amino acids, yes. Whole proteins, no. If somebody has a substantial amount of protein in their urine, that's a sign that they have kidney damage, and those holes have been opened up too large and for some reason, usually due to high blood pressure. So this is an even further zoomed-in view of capillaries. So that's an illustration of a capillary in a glomerulus. The openings or the holes in the capillary are called fenestrations. And so molecules have to be small enough that they can squeak through those fenestrations. And then furthermore, these big cells called podocytes, podo because it looks like they have little feet coming off of them, they form filtration slits. That's an electron micrograph view of what they actually look like. And so for water and the molecules dissolved in it to get out of your blood and become filtrate, they have to be small enough that they can squeak through those fenestrations and through the filtration slits that surround them. And if somebody has, chronic high blood pressure, eventually those holes can get stretched out and things like proteins can squeak through that aren't supposed to get through. Obviously, that's problematic. If someone has, acute kidney damage, like from a trauma, the capillaries can actually rupture and completely break. That's when you would start seeing blood in the urine, potentially. All right, this is a bit of histology, and even if you're not, beyond just the anatomy, this is important for understanding renal physiology. So this is a renal cortex. The easy way to know that it's a renal cortex is that's a glomerulus. That ball of capillaries plus the white space that surrounds it, that's the glomerulus, and then the capsule and capsular space that surrounds it. Right? These tubes are sections of renal tubule. It's difficult to say exactly what section they are, but it has to either be PCT or DCT because those are the sections of the tube that are in the cortex. Right? If we go further zoomed in, that's an even closer view of a glomerulus. You can see capillaries, all throughout here. That really, again, is just a big ball of capillaries, and then some of those cells, like the nuclei that you're seeing there, probably belong to podocytes. Remember those cells that are forming filtration slits around those capillaries? Okay. Obviously, this is the capsular space, so that's where fluid would be leaving those capillaries, and at that point, you would call it filtrate. The goal is for filtrate to start accumulating inside that space. Eventually, it's going to be pressurized, and it would pretty quickly leave through the PCT. I'm fairly certain that that's PCT, even though you can't see exactly where it connects to the glomerulus because it's somewhere out of the plane of the cut, right? And that little guy right there is probably the afferent arteriole, right? Blood flow coming in. Again, you can't see exactly where the connection is because it's outside the plane of the cut. All right, this view is renal medulla. No glomeruli present, all tubes. And these big guys, there and there and there, those are collecting ducts. Remember, multiple nephrons feed into one collecting duct, which is part of the reason why they're so much larger. The rest of these tubes are parts of the renal loop, either ascending or descending. For the most part, these thicker tubes that have cuboidal epithelia, called thick segment, are part of the ascending loop, and these thinner ones with squamous epithelia, called thin segment, are part of the descending loop. Why the different epithelial cell types are there isn't a big deal for right now, but I might briefly mention it later. All right, so the next thing that we have to tackle is explaining how pressure is regulated inside the glomerulus. Because remember, this 180 liters a day number is like a ballpark average. We call that GFR, glomerular filtration rate. And the primary variable that you can regulate is pressure. Okay, so if you have more pressure in the capillaries, the blood vessels of the glomerulus, the more fluid is going to come out. Right, and these numbers, HP stands for hydrostatic pressure. That's just normal blood pressure. And obviously that's pushing out of capillaries into that capsular space. So this number, 55 millimeters of mercury, is actually very high for a capillary because those capillaries need extra pressure to push fluid out. That's not happening anywhere near the same degree anyway in other organs, right? This osmotic pressure number is the same osmotic pressure you saw back in cardio when we were going through capillary beds, and I said that serum albumin proteins create osmotic pressure back in the blood. Same thing. Don't worry about explaining it for the exam. It exists. And then hydrostatic pressure pointing back into the glomerulus because filtrate accumulates in that capsular space and it pressurizes a little bit, and so to some degree that fluid fights back. All that really matters is this number, hydrostatic pressure, you can change. These two numbers, not so much. And so if you add them up and do net pressures, or subtract them, I guess, you've got 55 millimeters of mercury pointing out of those capillaries, and then 45 pointing back in, right? 30 plus 15 is 45. That gets you a net pressure of 10 millimeters of mercury in this direction. And so if the net pressure is pushing in this direction, that's the direction that water is going to move, right? And that number, net filtration pressure, that's by far the biggest variable that controls how much fluid actually leaves the blood and enters filtrate. That's the biggest thing that determines your GFR. Okay? And the way that we can manipulate this is this afferent arteriole can constrict and dilate. If you want hydrostatic pressure and net filtration pressure to go up, you would vasodilate the afferent arteriole and let more blood in. It's just like opening up the tap on a faucet. And if you want pressure and filtration rate to go down, you would vasoconstrict the afferent arteriole, which is like closing off the tap on a faucet. Should be pretty straightforward. Okay. So biggest variable, net filtration pressure. The other factors that affect GFR, surface area of capillaries, they're not really drawn or shown in most of these illustrations, but all of these capillaries have tiny bits of smooth muscle around them, and there's a hormone called atrial natriuretic peptide, or ANP, that can affect the surface area of those capillaries by relaxing or contracting those little bits of smooth muscle. We'll get to that later. That plays a secondary but still important role. The last one, the size of the holes in the membrane, that should not change. Okay? So that matters, in that if you have kidney damage or chronic hypertension, that number can get thrown off. but in a healthy person, that shouldn't be a variable. So important variable number one, net filtration pressure. Secondary variable, surface area affected by A and P. Again, we'll get there before too long. All right, so now that we've got the concept down that changing the afferent arteriole, constricting or dilating, will have a big impact on net filtration pressure and GFR. This slide is showing you two different mechanisms of how that control actually happens. Okay, so the gist is you normally want GFR to be fairly stable. It's not a perfect, always the same homeostasis, but pretty close unless pretty extreme things happen. So this concept, auto-regulation of GFR. Auto, think self. The kidney is self-regulating GFR, and the stimulus is a change in systemic blood pressure. And so the idea is, imagine that you go from sitting down and not doing anything to getting up and going to the gym and exercising really hard. Your systemic blood pressure is going to go way up. If you then quit and go back to resting, systemic blood pressure is going to go way down. So when those big blood pressure changes happen, if you weren't careful, you would have big surges or drops in pressure in the afferent arteriole. And so the concept here is the afferent arteriole has to constrict or dilate as needed to keep pressure in the glomerulus stable, even when your systemic blood pressure flies up and down. Okay? So they're using the example of pressure going down. You could easily reverse all this and do it for pressure going up as well. So if your systemic blood pressure is down, blood pressure in the afferent arteriole is also going to be down, which will make your GFR go down. You don't really want that to happen. And so if systemic pressure is down, the way that this is monitored is stretch receptors in the wall of the afferent arteriole will realize that they're not being stretched as much. Right? That's the trigger that tells them that pressure's down. If you want to get GFR back up, you've got to let more blood into the glomerulus. The result is vasodilation of that afferent arteriole, which will increase your GFR. It's really important to remember some stuff from cardio on this. Remember the difference between systemic pressure and local pressure. The initial problem is a systemic blood pressure shift. What we're regulating in the kidney through vasodilation is local pressure. And so if you vasodilate, that will increase local pressure in the glomerulus, right? Even though, remember, vasodilation decreases systemic pressure, it increases local pressure right on the other end of that arteriole, right? And this whole thing, like I said, could be flipped. If the problem with systemic blood pressure is too high, you would just switch all these down arrows to up arrows and say that instead of dilating, you would vasoconstrict instead, right? To protect the glomerulus from hyperpressurizing. All right, and the end goal is homeostasis of GFR. Okay, this other mechanism, the tubuloglomerular mechanism, I'm going to circle back to later because I have to go through a bit more microanatomy of the renal tubule before that's going to make a lot of sense. Okay, so again, autoregulation, self-regulation. This is the kidney effectively taking care of itself, even if there are big changes in systemic pressure. Okay. All right, this slide, which is really very important for this exam. This hormonal stuff is a really big part of what the kidney does to control blood pressure. And there are definitely going to be multiple questions pertaining to the next few slides on your test. So again, initial problem, systemic blood pressure is down. Hormonal regulation, if it's an endocrine hormone, the effects are going to be systemic. So autoregulation was the kidney taking care of itself. This is the kidney taking care of systemic blood pressure, which is going to have an indirect effect on GFR. So we'll say hormonal regulation of GFR and blood volume / mean arterial pressure. All right, so, and again, in their example, systemic blood pressure is low. Your kidney has sensors in it that know that pressure's down. And if pressure is low enough that the heart isn't really compensating, your kidney assumes that blood volume must be low. And so I want to just add a caveat on this. We're going to say that this is a decrease in systemic blood pressure specifically due to low blood volume. All right, if it's a heart issue, if your blood pressure is low because cardiac output's down, well, your heart has to fix that problem. But in this example, we're going to say blood volume is low. The kidney responds by releasing an enzyme called renin. Renin converts a pre-hormone called angiotensin 1 into the active form angiotensin 2. You don't really have to worry all that much about renin for me. Angiotensin is where all the function's at. So angiotensin is a systemic hormone, has two effects that lead to the same result. Effect number one, angio means blood vessels, tensin, like tension, to contract. Angiotensin causes systemic vasoconstriction, and this is important. This is not vasoconstriction of the afferent arteriole like we were doing back here, right? This is a local thing. It's autoregulation. This is systemic. You're constricting arterioles all throughout the body. The goal is get blood pressure back up because you know that systemic vasoconstriction increases mean arterial pressure, right? The problem is our initial issue was blood volume, okay? Low blood volume is a problem because low blood volume leads to low blood pressure, but this vasoconstriction, it solves the pressure problem, but it really doesn't solve the volume problem. And so I say this is a short-term solution, a short-term fix. You can vasoconstrict, you can get pressure back up, but the problem is vasoconstriction increases resistance. You don't want to walk around like that for the rest of your life. And so this is really a very temporary solution to a problem, but eventually you're going to have to actually solve the root problem, which is the volume issue. To do that, angiotensin will stimulate the release of aldosterone from the adrenal cortex. Aldosterone does a very specific thing. It increases sodium reabsorption, specifically in the DCT, that distal convoluted tubule that we'll see more of later. You have no way to pump water, but you can pump sodium. And since water follows salt by osmosis, if you pump sodium back into the blood, so when this says sodium reabsorption, that means reabsorbing it back into the blood as opposed to letting it out in urine. Water follows salt, blood volume goes up, blood pressure goes up. That blood volume increase, that's your long-term fix, right? That actually really gets you back to homeostasis for the volume issue, which was your root problem. Right. In order for that to happen, though, if your blood volume is going to go up, the water had to come from somewhere. Obviously, that's from food and drink. So for that reason, angiotensin also acts on the brain, on the hypothalamus, to increase thirst, because if you're going to get your blood volume back up, you're going to have to drink some water along the way. And that's why low blood volume will lead to a thirst response, and hopefully you'll seek out some water if it's available. All right. Okay, in all of this, don't forget, we haven't really gotten to the hydration part of this, but don't forget the difference between blood volume and hydration. And all the way back in the endocrine chapter, I drew a simple 4-square box on the board that said I had hydration on one axis. So this was dehydrated, this was overhydrated, and then I had blood volume on the other axis, and this was blood volume too high, and this was blood volume too low, right? The difference between hydration and volume is really, really, really important for this exam. Remember, hydration is measured in milliosmoles. It's basically saltiness. How much salt and protein and other molecules are dissolved in your blood. Blood volume is very simply measured in liters. how many liters of fluid are inside of you. And they're completely independent from each other. All right? So, angiotensin and aldosterone are entirely a volume issue. They don't technically have anything directly to do with hydration, right? So volume, angiotensin, aldosterone, and also ANP, which is coming up next. Hydration, this is controlled by antidiuretic hormone, ADH, which we'll get to eventually. But just to remind you of this stuff. So if the ones that make obvious sense, if you're dehydrated and your blood volume's low, that's this quadrant, that just means you're doing some combination of not drinking any water and maybe sweating a little bit. So if you're losing water, you're becoming low on blood volume and you're becoming dehydrated because the salts that remain back inside of you are becoming concentrated. Okay? Because remember, dehydrated really just means too salty. All right, this one, increased blood volume and dehydrated. If you have very salty food plus water, the salty food made you dehydrated and the water increased your blood volume. And so you have too much blood and it's very salty. That's high blood volume dehydrated. High blood volume overhydrated, this is if you just drink a bunch of water but don't have any or at least much salt. Again, the no salt part plus the water is making you overhydrated because you've got too much water relative to your salts. And again, the water is increasing your blood volume. Okay. The lower right-hand one is a bit more strange. Overhydrated and low blood volume. This really only happens when you have either blood loss or you sweat a lot. Like imagine somebody running a really long race. And then you only take back some fluid, but not quite enough fluid to get your blood volume up to homeostasis. So if somebody ran a race and they lost like 5 liters of sweat, but they only drank 2 liters of water, their blood volume is still low, but the fact that they're only drinking water and not salts is making them overhydrated. Okay, so again, treat these things completely separately. All right, this stuff, entirely a volume issue. This is the response to low blood volume. All right. The next slide, same thing. This is still a volume issue, but this is the response to high blood volume. And the response is releasing a hormone called ANP, atrial natriuretic peptide. The term natriuretic is supposed to mean like natural diuretic, because a diuretic is something that makes you lose water. And the concept is if your blood volume goes up, your blood pressure goes up, that is sensed by your atria in the heart because they get stretched out and pressurized by the extra blood volume. The atria release ANP. ANP targets smooth muscle cells in the glomerulus. Right? This is what I mentioned back here. If you relax the smooth muscle around those capillaries, you'll increase surface area. And if you increase surface area, you'll let more fluid out. And so the end effect of ANP is to increase GFR. And if you increase GFR, you're going to get more urine formation. And if you get more urine formation, you're going to get a decrease in blood volume because the water, by definition, is leaving your blood and entering urine. So blood volume goes down. If blood volume goes down, blood pressure goes down. Okay, so on the exam, if I ask about systemic blood pressure and volume being too low, you want to go through angiotensin and aldosterone. If we talk about blood pressure being too high and blood volume being too high, you want to go through ANP. All right, I think I'm going to stop here and make this part 1, and then I'll make part 2 and post that soon as well.